This video is meant to explain the concept of epistasis to someone without a biology background. To do this, we will be using a practice example in order to demonstrate how multiple genotypes could lead to the same phenotype. In order to provide some background information, we will briefly discuss Gregor Mendel's dihybrid pea cross. Mendel crossed pea plants that were homozygous for two genes at each loci had the same two copies for the trait. These copies are also known as alleles. He then self-crossed the heterozygous progeny, meaning he bred them with one another. He found that the genes assorted independently from one another, and that their genotypic and phenotypic ratios were therefore predictable, 9:3:3:1. His findings are now known as the laws of Mendelian inheritance. It is now understood that Mendel's rules of inheritance do not always apply, and that we sometimes observe fewer phenotypes than genotypes. When the expressions of alleles at one locus masks or modifies the expressions of alleles at another locus, we call it epistasis. In order to illustrate this principle of epistasis, we will be reviewing a fictitious case of single recessive epistasis in German Shepherd coat pigmentation.
So for the fictitious pathway I've created will be revolved around German Shepherd coat pigmentation, and this is a single recessive epistasis event. So here our individuals with dominant A's and dominant B's will be black because the dominant A gene is producing black pigment and the dominant B is suppressing tan bellies. So we will only observe black individuals with this genotype. Conversely, individuals with dominant A's and homozygous recessive B's will be black and tan because we are getting this black pigment production, but their tan belly suppression it's being expressed in this scenario. So we have black and tan German Shepherds. And the last scenario with our homozygous recessive A's, they are epistatically masking the B locus in this example. So we will actually only be observing white German Shepherds with homozygous recessive A's regardless of the B locus. And so now we're going to take a look at what this will mean for these crosses right here in terms of their genetic and phenotypic ratios. So I've gone ahead and drawn out the crosses respectively above and below. As you can see here, this kind of looks like Mendel's dihybrid cross. So we are observing 9 German Shepherds that are black right here in the blue. And we are observing 3 that are black and tan right here with this dominant A and homozygous recessive B's. However, these 4 right here with the homozygous recessive A's are actually exhibiting white coat color. And that is because the A locus is epistatically masking the B locus. So we are only observing white in these individuals. So our genotypic ratio, although it is 9:3 to 3:1, we are observing a 9:3 to 4 phenotypic ratio. And you can see here in the other cross, we are actually observing a little differently. We're having an 8:8 genotypic and phenotypic ratio because of our dominant A's and B's, which are taking up our left side of our dihybrid cross right here, and our homozygous recessive A's along with their dominant B's. We don't care what these B's are doing because our homozygous recessive A is telling us that those German Shepherds will have a white coat color. So their phenotypic and genotypic ratios are the same.